Hello, could somebody confirm you can hear me?
I can hear you.
Thank you. Sorry for being a bit late. I had to log into Zoom and it wanted verification code. And the first time the verification code didn't work, so I had to do it again. Let's see, where's our syllabus? There it is. All right, today is August 22nd. Let me share my screen. Don't get that. I'll say open the screen all the way. All right, you should be seeing my screen now. Not there's anything to show here, but let's actually go back. Can use this for the lab. There we go. Today is August 22nd. Go ahead and get my phone away. And today we are supposed to be working on chapter 13, viruses, viroids, and prions. Do note that we have a quiz tomorrow. It'll be covering chapter 8 and lab 9. Any questions about any of that? All right. Let's go on with chapter 13.
I have a question.
Uh-huh.
What is final A? It's on the calendar.
You have two finals, final A and final B. Final B will be the main one, having the multiple choice questions. You have to use Respondus lockdown browser to access final B. Final A has a few questions, and they are short answer or essay. You do not need to use Respondus lockdown browser for final A. And it's not timed either. It must be turned in within the day of the final, but you can spend 24 hours on it if you want.
Okay, thank you.
Okay. All right, I have here that we were talking about viral multiplication and bacteriophages. Is that correct? Anyone? We talked about the lytic life cycle and the lysogenic life cycle, two mechanisms that bacterial viruses, and they're called bacteriophages, can go through their life cycle and replicate.
Yes.
All right, great. I'd like a little bit of confirmation here. I know you guys don't like to talk, but it does reassure me that I'm on the right page. All right, let's talk about viral multiplication and bacteriophages. To make new bacteriophages, what do viruses need? What components need to be made in order for a virus to replicate? Okay, what do viruses have? Anyone? What molecules do viruses have?
Are you looking for DNA or RNA?
Yeah, they have a genome, which is either DNA or RNA. We'll talk a little bit more about that. They also have a protein coat around the genome. And so those molecules have to be replicated in order for the virus to replicate. It is true some viruses have an envelope, and we will talk about that much later today when we're talking about animal viruses. I'm trying to think, are there any bacteriophages that have an envelope? And I don't know of any, but I'm not a viral expert. So at the minimum, viruses need to replicate their genome and they need to make their protein coat. how they do that, if it's a double-strand DNA virus, it will replicate the DNA, and that's pretty easy. You just separate the DNA, use one strand as a template to make the other strand, and then you replicate your double-strand DNA. Then you have to transcribe the DNA to make the viral messenger RNA, and then the viral messenger RNA will be read by the ribosome to make the capsid proteins, the viral proteins. And then the viral proteins have to get together with the viral genome to make the new progeny virions. This is a minimum of what we need in order for the virus to replicate. Any question about any of that? All right, let's talk about the lytic life cycle. The lytic life cycle happens in 5 stages. There's attachment, penetration, biosynthesis, maturation, and release. Let's watch a little video of the lytic life cycle.
First, the
Viruses.
phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage receptors. Next, the DNA of the phage is injected into the cell, as if from a syringe. The ends join, forming a circular molecule. Then, the injected DNA replicates inside the host bacterial cell, and phage heads and tails are synthesized. The phage DNA is then packaged into the heads, and the filled heads attached to the tails. Finally, the cell dissolves, or lyses, liberating about 100 newly formed phage particles after 45 to 60 minutes.
All right, this computer doesn't have VLC as its movie player. It only has whatever Microsoft provides. And with Microsoft Movie Player, it will not show the pictures in the movie. It only gives the audio. So let's go on to another one.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, the next part is the lysogenic life cycle, and I'll show that in a little bit. Any questions about the lytic life cycle? What do we have here? All right, that's good. So first, in the lytic life cycle, the virus has to attach to the host cell. The phage, the bacteriophage, sorry, I always try to use the word bacteriophage to make it clear that I'm talking about a bacterial virus. The bacteriophage attaches to the host cell with its tail fibers. We saw that in the movie. I don't know if I can show that, but let's go on to the next one. There we go. So the host cell is here and it's attaching with the tail fibers. These are the regions of the virus that have the adhesions or ligands that bind to the receptors on the host cell. So the receptor will be the molecule in the host cell, and then the tip of the tail fiber will be the ligand, or you can call them adhesions, which is kind of shown there, but this is easier to see. Next comes penetration, where the phage puts its genome inside the bacterial cell. How that happens is the phage releases lysozyme, which opens up a small hole in the cell wall, and then the tail sheath literally contracts like a syringe, and that puts pressure on the head of the virus, and that pressure will put the genome inside the head of the virus. It'll inject it into the cell. And there you can see the virus like that, and there it is contracted. And that puts pressure on the head, which injects the genome of the virus into the cell. Any question about any of that? Next, the virus takes control of the cell, and it uses the cell to produce bacteriophage DNA, because we're talking about a DNA virus, but it could be, if it's an RNA virus, it would make a viral RNA, the genomic RNA. And then also it makes the viral protein. So that's biosynthesis, followed by maturation. That is the assembly of the new virion, where the viral genome is put into the viral head and the proteins of the virus come together. It self-assembles. And then there's an enzyme that stuffs the virus with the viral genome. And then that's followed by release, where the phage, the bacteriophage releases the enzyme lysozyme. That's the last gene of the virus which is expressed. And that lysozyme goes out and dissolves the peptidoglycan in the cell wall, which weakens the cell wall. And eventually water will move into the cell, and then the host cell will burst. And when the host cell bursts, that releases the newly replicated virions. Any question about any of that? So the virus attaches to its host cell. It dissolves a little piece of the cell wall, and then the virus contracts. That will inject the viral genome in the head into the cell. The cell, one of the things it does to, I'm not the cell, the virus, one of the things it does once it's inside its host cell is one of the early genes which is expressed is a nuclease, and it cuts up the chromosome of the host cell. Why does a lytic virus do that? Because the lytic virus wants to call the shots in the cell. And an easy way for calling all the shots in the cell is to digest the viral genome. Not the viral genome, it digests the cellular genome. And then only your genome is capable of directing the activities of the cell. And what the viral genome does is it replicates itself in biosynthesis, and it transcribes itself to make viral messenger RNA, which goes to the ribosomes. If this is in well, we're talking only about bacteriophages, so it's already in the cytoplasm. And then the ribosomes transcribe the viral messenger RNA, making the viral proteins. Most of those viral proteins are the capsid proteins, of the protein code of the virus. The virus proteins self-assemble, and then there's an enzyme which stuffs the head of the virus with the viral genome. Any question about any of that? All right, where is that shown here? Looks like I need one more slide talking about the last stage. And the last stage is, like I said, the last gene of the virus to be expressed is the enzyme lysozyme. And that enzyme comes out and digests the peptidoglycan in the cell wall, weakening the cell wall. And then that cell will burst. And once it does, oh, there it is there. Once it does, that will release the newly replicated virions. So in the lytic life cycle, there's two terms you should know. The burst time, that's the time from the initial infection of the host cell by the virus to the time when the cell bursts. And this averages 20 to 40 minutes, depending on the viral species and the temperature that the virus is at. And then the burst size, that's the number of newly replicated new virions that are released when the cell bursts. This depends mostly on the species of the virus. It's usually between 50 to 200 newly replicated virions are released. And where do those new virions go? They go to neighboring cells. And then they infect them. Any question about any of that? All right. So that's the lytic life cycle, where when the virus replicates, it kills its host cell. Not only does it digest the host cell's chromosome, but it also bursts the cell. The lysogenic virus follows a different life cycle. Upon attachment of a bacteriophage to its host cell, once the genome of the virus is inserted into the host cell. So that happens similar. The virus will attach to its host cell, and then it will contract, injecting the viral genome into its host cell. But two things can happen once the viral genome of a lysogenic virus enters the host cell. The virus has to decide immediately whether it wants to engage in the lysogenic life cycle or whether it wants to engage in the lytic life cycle. If the lysogenic virus decides to enter the lytic life cycle, then the lytic life cycle will happen as we just discussed in the lytic life cycle, and the virus will replicate, digest the cell, burst the cell, and then the newly replicated viruses will be released. So the lytic life cycle of a lysogenic virus follows the lytic life cycle. And it's the same, except when the virus first enters the cell, it has to decide, do I want to enter the lytic life cycle or do I want to enter the lysogenic life cycle? If the virus decides to enter the lysogenic life cycle, the host cell will remain alive and it will begin a lysogenic life cycle for the virus. The lysogenic virus will remain latent, inactive, and the bacterial host cell is termed a lysogenic cell. It remains alive, during the lysogenic life cycle. So as long as the lysogenic life cycle continues, the host cell will be alive. Any question about any of that? All right, let's follow the lysogenic life cycle. Here it's here. Initially, the lysogenic virus attaches to the host cell. It penetrates, sending its genome into the host cell. The host cell has to immediately decide, not the host cell, the virus has to immediately decide whether to enter the lytic life cycle or the lysogenic life cycle. I've already talked about the lytic life cycle. It just follows the lytic life cycle normally. But once it decides to enter the lysogenic life cycle, the viral DNA will integrate into the host cell's chromosome and become a part of the host cell's chromosome. We call the viral DNA inside the host cell's chromosome a prophage. Now, the only time the viral genome can replicate is when the cell divides. When the cell will divide, it will replicate its DNA, and when it replicates its DNA, it will replicate the viral prophage. And then the two daughter cells each get the host cell's DNA as well as the viral DNA. And that is the only time the lysogenic virus replicates, when the virus is following the lysogenic life cycle. Any question about any of that? All right, the lysogenic life cycle can continue for many generations. And that's shown here. But every once in a while, in one cell of the progeny of this cell, and that could be many progeny, we don't know, but it could be many, one cell will leave the lysogenic life cycle and then enter the lytic life cycle. What happens is the prophage is excised from the chromosome and then becomes a separate viral molecule. And then the lytic life cycle proceeds, where the viral DNA will replicate, the viral proteins will be made, the viruses will self-assemble, the virus will degrade the host cell wall, the virus will burst, releasing the newly replicated virions. Any question about any of that? Now, what leads the virus to leave the lysogenic life cycle and enter the lytic life cycle is not well understood. They've been studying this for over 100 years, and scientists have never come up with an answer. Why does the lysogenic virus leave the lysogenic life cycle and enter the lytic life cycle? The only thing they've found where this can be encouraged is if you damage the host cell DNA, like expose it to radiation or something. The virus apparently recognizes that the host cell's DNA is damaged and that the host cell may die, and that will encourage the lysogenic virus to leave the lysogenic life cycle and enter the lytic life cycle. But other than that, why a cell leaves the lysogenic life cycle and enters the lytic life cycle is not well understood. Any questions about any of that? All right, let's talk about this in a little more detail. The viral DNA, which is in a circular molecule, will integrate into the host cell chromosome. We call that a prophage. There is a repressor protein that is expressed when the cell is in the lysogenic life cycle. The purpose of this repressor protein is to keep dormant the lytic life cycle genes. So all of the genes involved in the lytic life cycle are kept dormant by the repressor protein. And then the cell goes through cell division, replicate its host chromosomal DNA as well as the viral DNA. And that can happen for many generations. Occasionally, the prophage excised from the bacterial chromosome, and that will initiate the lytic life cycle. Any question about any of that? If not, let's watch the little movie on that.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any question about that movie? Everything is right on with this movie, with one exception. Let me see if I can show it here. No, I can't seem to show it. Ah, here it is. Go a little forward.
And a new generation of
There. It's showing T4 for the virus. And T4 is not a virus that's capable of entering the lysogenic life cycle. T4 can only enter the lytic life cycle. But other than that, you guys probably would have never caught it. That's the only mistake in this video. That shouldn't be T4. Any question about any of that? All right. There are some consequences of lysogeny when the virus is in the lysogenic life cycle. First, the host cell, when it is infected with a lysogenic virus, this cell is now immune to infection by the same bacterial virus. So it cannot be infected by another bacteriophage of the same species. So this cell is immune. Second, phage conversion can happen where the host cell can take on new properties. Why does this host cell take on new properties? Because it has new genes. The new genes are the viral genes, the genes of the virus. And that can change the phenotype of the host cell when those genes are expressed. The best known example of phage conversion is when a lysogenic virus infects Corynebacterium diphtherium. Corynebacterium diphtherium only produces the Corynebacterium diphtherium toxin when the bacteria is infected with a lysogenic virus, because the Corynebacterium diphtherium toxin is a gene on the viral genome. So the Corynebacterium diphtherium bacteria will make you less sick if you have an infection, and the bacteria is a bacteria without the lysogenic virus. On the other hand, if you get infected with Corynebacterium diphtherium and it has a lysogenic virus within it, you can get very sick because this cell will make the Corynebacterium diphtherium toxin. So yes, how sick you will become will depend on whether your bacteria is infected with a virus or not. Any question about any of that? All right. So enough about bacteriophages. Let's move on to talking about multiplication of animal viruses. Viruses that infect animal cells. Any questions before I proceed? The multiplication of animal viruses follows the basic pattern of bacteriophage multiplication, but there are several differences. First of all, there will be an attachment difference because animal viruses attach to the cell membrane. Animal cells do not have a cell wall like most bacterial cells. The penetration will also be different. The animal virus doesn't contract and inject its viral genome into the host cell. The penetration into an animal cell is much easier because it's easier getting through the cell membrane. You just simply enter the cell by endocytosis or by fusion. And I'll talk about fusion in just a minute. The encoding of the virus can be similar. Biosynthesis is pretty much the same too as in bacterial viruses. Maturation is similar. Release can be different. It can be the same, meaning there are animal viruses which follow the lytic life cycle, where the animal virus will infect a host cell, replicate in it, and then lyse the cell to release the lytic viruses that have been replicated in the cell. So we do have animal cells that follow the lytic life cycle, but we also have animal viruses that follow another life cycle, like the budding life cycle. There's also the persistent infection life cycle and the latent life cycle. So those are a couple of different life cycles, and they do not follow the lytic life cycle. Let's talk a little bit about the budding life cycle virus. The budding life cycle virus will replicate in its host cell, but only at low numbers. The cell won't make tons of newly replicated budding virus. And then the virus will slowly bud out of the host cell. When the budding virus replicates, it usually does not kill the host cell. If the host cell is killed, it's because the immune system recognizes that that cell is infected by a virus, and then the host immune system will kill that cell. The virus does not. Any question about any of that? Sorry, I'm not doing too well today. I came down with a cold last night. All right, multiplication of animal viruses follows the general pattern of viral multiplication of bacteriophages. There are a couple of differences. The main difference you need to know is that in bacteria viruses, they can only replicate in the cytoplasm because in a prokaryote cell or any bacteria cell, all there is the cytoplasm. That isn't the case in an animal cell. Animal cells have a nucleus as well as the cytoplasm. And the virus can either replicate in the cytoplasm or it can replicate in the nucleus. Any question about any of that? For the viruses that replicate in the nucleus, they are mostly DNA viruses. Why? Because DNA has all the machinery to replicate in the nucleus. So the DNA viruses replicates in the nucleus. The RNA virus, on the other hand, replicates in the cytoplasm of an animal cell. And why is because the cytoplasm has the ribosomes, the messenger RNA, the tRNA, and what else? Well, the ribosomes, the rRNA. So, and the RNA virus needs those to replicate. So RNA viruses tend to replicate in the cytoplasm. And this is known as a rule of thumb. A DNA virus replicates in the nucleus. An RNA virus replicates in the cytoplasm. With that rule of thumb, I should mention that there's one exception to the rule of thumb, and that is retroviruses are an rRNA virus, meaning their genome is rRNA, not rRNA, their genome is RNA, and they replicate in the nucleus. So retroviruses are the only known exception to this rule of thumb. And I'm talking about the viruses that I know of. I'm not an expert, so it's possible there might be other exceptions, but to my knowledge, the retroviruses are the only exception. They are an RNA virus, but they replicate in the nucleus. All right. The penetration or attachment of the virus in an animal cell is a little different because you're attaching to a cell membrane. However, you have the same molecules on the virus and the same molecule on the host cell that the virus binds to. So once again, the molecule of the host cell that the virus binds to are called receptors. And then the molecules of the virus that bind to the receptor are called adhesions or ligands, depending on who the author is. You know, I don't know which term Tutora uses, but those are the two terms which are used. And then penetration of the virus also differs. The virus can get into the cell by endocytosis or by fusion. Endocytosis is where the virus attaches to the host cell, and this region then invaginates, and then this membrane seals here, which will seal the cell membrane, and then that will make an organelle membrane around the virus, and then this organelle will be inside the cell. Any question about any of that? Anyways, that's endocytosis. I should mention, if you have an older version of the textbook, it incorrectly states that the virus can enter the cell by penocytosis. That is not correct. It is endocytosis. Oh, let me talk about fusion. Really best if I draw this one. So here we have the cell, and it has a lipid bilayer for the cell membrane. So I'm going to draw two of these. And there we have an envelope virus. So we'll draw the virus here. And then it's surrounded with a envelope, which is a double layer. So I'll try and make that a double layer. The envelope confused to the cell membrane. So let's move the cell up a little bit. Oops, that's not what I wanted. It's easier moving the cell than moving the virus. Taking a little while. There we go. So the virus binds to the host cell. And let's make the envelope this way of the host cell. The envelope confused to the cell membrane. Let me see if I can draw this. Yeah. Let's make it a little bigger. Oops. Now, the cell membrane doesn't go across like this. The cell membrane goes over here. It's fused to the envelope, and the cell membrane becomes a part of the envelope of the virus, and then continues. You'll note that when that happens, this virus can move into the cell through that hole there. And that's how the virus enters the cell by fusion. Any question about any of that? All right, biosynthesis, we talked about the virus has to replicate nucleic acid and proteins, and then the virus assembles in the step of maturation. But the replication of the virus can happen by more than the lytic life cycle. Animal viruses can engage in the lytic life cycle, where the virus will replicate in the cell, and then the virus will lyse the cell, and then that will release the newly replicated viruses. So that's the lytic life cycle of an animal virus. We also have the budding life cycle, where the virus replicates and then assembles. I don't think I can show you the viral genome in here, but I'll try. Up, what happened there? Didn't want to do that. Well, let's just go to slide. Yeah, I don't see it in there, but the viral genome is inside of this viral capsid. And then the virus comes up to a membrane. This can be the cell membrane, but it could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. Which membrane the virus uses will depend on what species of virus it is. Like for HIV, it's the cell membrane. Most viruses, I don't know which membrane it hits, but I do know for AIDS, it's the cell membrane. And what happens is the virus comes up to the cell membrane and then puts pressure on a region of the membrane that has viral proteins in it. And these blue dots would be the viral protein. You'll notice this region over here, as well as this region over here, which you can barely see, do not have the viral proteins in it. So this virus will not try to bud out here. It'll go where the viral proteins are and then try and bud out here. As it buds out, it puts pressure on the membrane, forming a bud, and then this virus will actually rip off the bud where what used to be the cell membrane will close on itself and form the envelope of an enveloped virus. And then right here, the cell membrane will reform, meaning this pinches off, and then this, instead of going this way, will actually go connect to there, and this one will connect to there. So the cell membrane is reestablished in the budding life cycle. Usually the budding life cycle virus does not kill its host cell. The budding is happening at low enough numbers so that the cell losing a little bit of membrane is not a concern to the cell. Here we're looking at the budding life cycle in an actual electron microscopic image, and there is a cell, and you can see several buds on it where a virus is replicating on the hepatic liver cell. Any question about any of that? Oh, we can't move that forward for some reason. All right, viruses have different genomes, so let's talk a little bit about how the different genomes replicate. Viruses can have double-stranded DNA for their genome, and that's pretty easy to replicate. You just separate one strand from the other, and then you use one strand as a template for the other strand, and that will replicate the other strand of DNA. Any question about any of that? Single-strand DNA is not unheard of. Our cells have single-strand DNA whenever a gene is being transcribed or whenever a protein is being made. But at all other times, the retroviruses are a family of 2 viruses. Any question about any of that? All right, let's talk about the RNA virus genomes. Viruses can have double-strand RNA for their genome. I think I mentioned this before. This is unheard of in eukaryotic cells, actually any bacteria cell as well. And that is, we do not have double-stranded RNA, but a viral genome can have a double-stranded RNA molecule for its genome. Any question about any of that? And then there's single-strand RNA, which can be the cell's genome. Whenever you're talking about single-strand RNA, you have to mention, are you talking about positive strand, single-stranded RNA, or are you talking about negative strand, single-strand RNA. Any question about any of that? So with the single-strand RNA, you have to distinguish whether the single strand is the positive strand or the single strand is the negative strand. And it's usually abbreviated ssRNA, which is single-strand RNA. And the replication happens depending on what the genome is. So each virus type needs to have a slightly different mechanism for multiplication and biosynthesis of its genome. For the DNA viruses, this chart is showing you that there are 7 families of DNA viruses that cause human disease. Now, do understand there are more viruses. However, in animal cells, the ones that are pathogenic to humans are shown here. And if they're DNA, they belong to one of these 7 families. You don't need to know the families. We will talk a little bit about poxviridae and herpes viridae. All the others we won't talk about much, although adenoviridae I may talk about one virus. The table does show you the type of DNA it has for its virus, whether the virus is enveloped or naked, and then there's complex symmetry and its size. And I'll be mostly talking about the herpes virus, I think. So with adenovirus and herpes simplex virus, both of which are DNA viruses, a virus that has DNA in its genome, the DNA is generally copied in the nucleus using viral enzymes. So of DNA viruses, they're usually copied in the nucleus. And I don't know of any exceptions. A DNA virus that does not replicate in the nucleus. I'm gathering that there are, but I don't know of any. So it's not very common, and the log would be very long. So once the DNA enters the host nucleus, it has to decide, is the DNA going to become a provirus, where the genome of the virus becomes DNA, and then DNA is permanently a part of that host cell's DNA. So when an animal virus enters a host cell, if the animal virus ever joins the chromosome, then the chromosome will always have that DNA virus. That there's no other way you can have the antigens of a virus in the cell. And the viral genome stays in that chromosome for the life of that host cell. And the provirus, which is the viral DNA in the host cell of an animal cell, we call it a provirus, not a prophage. The only time that provirus is directly replicated is when the cell's DNA replicates, making 2 chromosomes, one going to each side of the cell, and the cell splits down the middle. The point is that each new daughter cell has a chromosome that has the provirus in it, the DNA coding for a lytic life cycle virus. The other thing that can happen once a viral DNA animal virus enters its host cell, the viral DNA can be expressed, producing new virions. Shown here, making new virions. And then the new virions will be released from the cell, either by the budding life cycle or by the lytic life cycle. Any questions about any of that? If not, let's talk about a virus, which is a DNA virus, and how it replicates in its host cell. The virus will attach to its host cell using receptors on the host cell and ligands or adhesions on the virus. The virus will then enter the cell either by endocytosis or by fusion of the viral envelope to the cell membrane. Once the viral DNA is inside the cell, viral messenger RNA will be made, which will then be copied, and then the viral protein will be made from the viral messenger RNA. With the viral genome, it depends on whether the virus is DNA or RNA. We'll just say double-stranded DNA because it's so easy to replicate that. I'll talk about that as my example. And then the viral genome will be replicated, and you just split it into two strands. You will use one strand to make the other strand. And then there will be assembly of the viruses. Oops, too far. Too far again. So the viral DNA will be replicated, and then the viral DNA will be transcribed to make viral messenger RNA. The viral messenger RNA, if need be, will move into the nucleus. And then the viral proteins will be made, mainly the capsid proteins. And the viral genome will be exposed to the viral capsid proteins, and they will self-assemble into new virions, which will then either lyse the cell to release the newly replicated virions, or if this is a budding life cycle virus, the virus will bud out of the cell at low numbers. Whatever route the cell takes, the new virions will be released, and the cells that they will be most likely to infect will be neighboring cells. Any question about any of that? All right, here we have some animal viruses. And you'll note there are more RNA families of RNA viruses or animal viruses, oops, too far, that cause human disease than there are DNA viruses. There are 15 families of RNA viruses that are known to cause disease in humans. And they're shown here. You don't need to memorize this table, but it does tell you the family the viruses are in, the DNA strand, what the genome is. whether the virus is enveloped or a naked virus, the capsid symmetry, the capsid size. As I mentioned before, the smallest virus you're probably aware of is poliovirus, which I'm not seeing where that is here now. Well, just skip it. You don't need to know about the capsid symmetry, the size, and then just some representative genera, like of the Picornaviridae, we have the poliovirus. We also have the viruses called rhinoviruses that cause the human colds, and then hepatovirus, hepatitis A, is also a Picornaviridae. The Picornaviridae is a very large group, and they're more diverse than most other viruses. Like hepatitis A is nothing like the virus that causes the human colds, the rhinoviruses, and then the disease polio. All very different diseases. With the viruses causing the human colds, remember there are 3 viral families that cause the human cold. The number 1, meaning the family of viruses that cause most of the human colds, belongs to the family rhinovirus, or family Picornaviridae. The number 2 is actually in the family Coronaviridae. And the number 3 we mentioned is the Adenoviridae. There are strains of Adenoviridae which cause the human cold. Any question about any of that? All right, we'll talk a little bit about Coronaviridae and Retroviridae. Most of the other viruses I'm not going to talk about any more than I already have, and if I do, it'll be an exception, and I'll mention this as an exception. Here are some RNA animal viruses. You can note that the viruses take on many different shapes and forms. Excuse me. All right, so the multiplication of RNA viruses follows the same pattern of the multiplication of the DNA viruses. Obviously, there are some differences because RNA will be the virus's genome, not DNA. Viral replication, as I stated before, as a rule of thumb, happens in the cytoplasm for RNA viruses. There is one exception, and we'll talk about that a little bit later today. The retroviruses are an RNA family of viruses, but they replicate in the nucleus, not in the cytoplasm. When we're talking about viral RNA replication, it's important to mention whether we're talking about the positive strand RNA or the negative strand RNA. The positive strand RNA, such as an animal virus, these strands of DNA can be recognized as foreign by the differences in the genes transcribed from the positive or the negative strand RNA. So for a DNA virus to be replicated, all you need to do is separate the two strands of DNA and then use one template to replicate the other. It's a little more tricky with a single strand RNA DNA virus. Let me go through that now because I don't have that on that slide here. So single strand. Single strand DNA virus replication. We have one strand of DNA. Come on. How we replicate it. I'm still not doing that. Come on. If this is the positive strand of DNA, how it will replicate is it will make the negative strand DNA well, that's not a very good arrow. Well, we're going to go with it because I don't have an easy way. So it'll make the negative strand DNA. And then use the negative strand DNA to make more positive strand. Let me see if I can blow this one up. Yeah, that's better. Up. Didn't mean to do that. There we go. And if the DNA was single strand, let's go ahead and do that now. I'm going to erase that. You just erase that. Make that single strand DNA. That's sort of out of place, but maybe I move it. Oh, come on. Ah, well, having a hard time moving this, so. Oh, well, we're going to leave it there. And then this will become positive strand DNA. Let's see if I can just draw that in there. Sort of. And then that'll be negative strand. Maybe I can just erase that. Oops, too far. No, I'm not going to try that. It's too difficult. Any question about any of this? Oh, come on. There we go, kind of. So that's how the single strand DNA virus replicates. For the single strand RNA, maybe just erase that. have a real hard time getting the R in there, I'm sure. Let's move that up a little. There we go. Ah, well, close enough. Told you I'd have a hard time. You just switch that to RNA, and I'm gonna have a hard time doing that. So oh well. And then this will be an R over here, meaning the single strand RNA replicates by making the negative strand RNA replicates by making the positive RNA strand, and then the positive strand RNA will be replicated to make the negative strand RNA. Let's erase that. So the single strand RNA replicates similar to the single strand DNA. Now the double-stranded RNA is really easy to replicate. You have two strands of RNA. And what you do is you separate them, and then you use one strand to replicate the other strand. Maybe I can do that with an arrow this way. So if this is the negative strand here, then this will be the positive strand of RNA. And if this is the positive strand, I'll try and draw that over here. You use that one to make the negative strand of RNA. Any question about that? And that's very similar to how double-strand DNA replicates. All right, let's go back to here. For DNA, the DNA will be transcribed to make the viral messenger RNA, and then the viral messenger RNA will be read by the ribosomes to make the viral protein. For the positive strand, single-strand RNA virus, it can be used by the ribosome as viral messenger RNA, and it can be translated into the viral protein. That's not the case for the negative strand RNA genome virus. It cannot be read by the ribosome. It does not have the code AUG, for example. It has the complementary code. And so the ribosome cannot read the negative strand, single strand RNA genome. But what the negative strand virus does is it makes a positive strand RNA molecule from the negative strand. And then it uses that positive strand to make more of the negative strand to copy the viral genome. And then it also uses that positive strand RNA as viral messenger RNA to make viral protein. Any questions about any of that? All right, let's go through viral replication. In an animal cell that has a negative RNA virus genome, the negative RNA genome will replicate into a positive RNA strand, and then use the positive RNA strand to replicate more of the genomic negative strand RNA. The positive RNA strand can also be used by the ribosome to translate the viral proteins, to make the capsid proteins needed by the virus, and then the capsid proteins will get together with the replicated negative RNA genome to make the new progeny virions. For a positive single-strand RNA virus, it will replicate by making a negative RNA strand of the virus, which will then be used to make the positive genomic single-strand RNA virus. And then this positive strand can be used by the ribosome to translate the viral proteins, mainly the capsid proteins, which will then get together with the replicated viral genome to make the progeny virions. Any question about any of that? So for a positive strand RNA single-strand virus, its multiplication will happen in the usual way, where the virus will bind to the host cell, get into the host cell, the RNA will can be read by the ribosomes immediately to make viral protein, and then the viral positive strand RNA will make a negative strand RNA, and then the negative strand will be used to make more of the positive strand RNA virus, which is the replicated viral genome. The replicated viral genome will then get together with the capsid proteins to make the new virions, which will then leave the cell either by budding or by lysis, if it's following the lytic life cycle, to leave the cell and then start the cycle over again. Any questions about the replication of positive sense single-strand RNA virus? If not, let's talk about negative sense single-strand RNA virus multiplication. The virus will attach to its host cell, get inside the host cell. The virus is negative strand, so it cannot make viral proteins. It first has to make the positive strand RNA, which will be used to make more of the viral genome, the negative strand. The positive strand RNA can also be used by the ribosomes to make the capsid proteins and other viral proteins, which will then get together to make the new virions. And then the virions will be released either by budding or by the lytic life cycle, lysing the cell, and then the cycle will continue. Any questions about the replication of animal viruses, single-strand and RNA animal viruses? All right, if not, let's talk a little bit about retroviruses. Let me make sure. I think I'm out of time, although this is the summer, so I might have more time. Yeah, we go to 6:50, so I do have more time. Let me talk about retroviruses. Here it is there. Retroviruses belong to the family Retroviridae. They include the lentivirus, which includes HIV-1, HIV-2, which cause AIDS, and another lentivirus is feline leukemia virus, which causes a kitty disease very close to kitty AIDS. The difference is the cat's immune system isn't killed by the virus, it's killed by the feline leukemia virus, which causes leukemia in the cats, and the leukemia gets so bad that the cat's immune system is shot to hell, and then the cat gets multiple infections and eventually will die of feline leukemia virus. Any question about any of that? Retroviruses are unique because they contain a gene for the enzyme reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase, and it makes DNA from an RNA template. And retroviruses is a name that actually comes from a shorthand of reverse transcriptase. You take RE from reverse and TR from transcriptase, put them together, add the O, and you have retro for retroviruses. There's a retrovirus, and you can actually see the reverse transcriptase, a protein in the virus itself. What happens is when the virus infects a cell, the genome of the virus gets inside the cell, which is positive strand RNA. Single strand. Positive, oops, it's not a positive. Positive single strand RNA virus. And it has reverse transcriptase in it. So let's put the arrow there going that way. Oops, see if I can rotate that. There we go. And we need RT for reverse transcriptase. What it first does is it makes a single copy of DNA. We'll go ahead and do that. Oops, a little bad. So this is a single strand DNA. And then in a second step, let's see if I can copy this. I did. Now let's see if I can move it. Reverse transcriptase will then work on the single strand DNA to make double strand DNA. So the making of double strand DNA happens in 2 steps. I think your book doesn't mention that, but it's not big deal. So there, from RNA, we get double-stranded DNA with the help of the enzyme reverse transcriptase. You'll note that this is not the way the DNA normally flows, the information normally flows in a cell. The information here is flowing from RNA to DNA. And in the cell we mentioned earlier that information always flows from DNA to RNA to protein. Well, in reverse retroviruses, the information does flow from RNA to DNA. And that happens because of the enzyme reverse transcriptase. Any question about any of that? All right, this double strand DNA, then, let me see if I can do that again. Then, a little bigger, but maybe I can make it smaller. Then integrates into the host chromosome. This integration is fairly random, meaning it can happen in different places, different chromosomes, and different cells. once that DNA enters the chromosome, it is permanent for the life of the cell. And you can see that when HIV infects a human, you never know when you might be cured of HIV, because at any time the virus can come out of the chromosome, meaning there have been a few people that they think have been cured of HIV, and you never know, because the cell could have the HIV DNA integrated into the host chromosome, and that will last for as long as this cell lives. The only way to permanently be cured of HIV would be for all of these cells to be dead. And you just don't know when that would ever be the case. You only need one for the virus to actually come out of the chromosome and then begin replication again, and then you could get HIV again. And that's just one of the problems of HIV. This is a minor problem, because if you ever get HIV to go in remission, and then you say, I might be cured, that would be really good news, even if you're not cured and it comes back 5 or 10 years later. Still, that would be very good for an HIV patient. But like I said, there are a few patients that they think have been cured of HIV, and as far as they can tell, they had no retrovirus replicating in them. Like I said, the problem is that the virus can come out of this chromosome, and what happens is, let's go ahead and do that again. Do it here. Normally, the RNA can be transcribed and what this is positive RNA. And then that can be translated and that will be viral proteins. So they can be translated from the RNA, and then these viral proteins can get together with the, let's see if I got one going sideways. No, well, yes I do, right here. Oh, don't have the right thing. And then get together to make the new virions, meaning the viral protein gets together with the copied viral RNA, and then you make the new virions. And they will bud out of the cell. Should go right. There we go. And then that will make new virions. Any question about any of that? So let's go with a slide. The biosynthesis stage of a retrovirus differs, and that is the positive RNA will use reverse transcriptase in 2 steps to make double-stranded DNA. First, it makes single-strand DNA. And then the double-stranded DNA can be transcribed into the viral genome. That's positive RNA, so it can be translated in capsid proteins. Capsid proteins can get together with the viral genome to make the new progeny virions. So a virus can attach to a cell, get inside the cell. The viral RNA will use reverse transcriptase to make first single strand viral DNA, and then the reverse transcriptase will make double-stranded viral DNA. But viral DNA will then integrate into the host chromosome, and that happens fairly randomly. It could mutate the cell, actually, but we're just going to call it a provirus for now. And that's permanently a part of this cell, that viral DNA in that cell. And then the virus can be transcribed. That's the viral genome, positive strand. It can be read by the ribosome to make viral proteins. The viral proteins can get together with the viral genome to make the new virions. And for HIV, that will bud out of the cell. Any question about any of that? All right, that's it for retroviruses. I wonder if we have time for coronaviridae. See if I've got time on here for tomorrow for retroviruses. I do. Let me see how much time I have and how many slides I have left. 65. And I'm on 50. I probably should go on and talk about coronaviruses. I'll try and be quick about coronaviruses. Coronaviruses make coronaviridae. Here you're looking at coronaviruses, and it was actually a slide that I chose to make the course. And I did this many, many years before COVID-19 ever came out. I just thought it was a cool looking virus, and so I chose it for the course, and that's what we have, which this is family Coronaviridae. This is not actually COVID-19, because when this picture was taken, COVID-19 did not exist yet. Coronaviruses are single positive strand RNA viruses. They are enveloped. Coronaviruses are actually the second most common cause of the cold. So coronaviruses have been known for a long, long time. They are the second most common cause of the human cold. They also cause other diseases like COVID-19, SARS, and MERS, which all of these three are similar to each other. They give severe acute respiratory syndrome. That's what SARS stand for. And then MERS, Middle East Respiratory Syndrome. MERS has the highest mortality rate. About 36% of the people who come down with MERS die. Fortunately, MERS does not spread from patient to patient. All of the people who've gotten MERS, at least that I'm aware of, are people who have a lot of contact with camels. Apparently, MERS is a virus that lives in camels, and it can grow in people, but it does not spread from person to person. So it needs the camel, apparently, to really replicate in, although it can infect you and make you sick, and it can kill you. I mean, 36% of the patients die. So fortunately, this disease does not spread person to person. MERS has a higher mortality rate. About 10% of the people die compared to COVID-19, so higher mortality rate. Fortunately, though, not MERS, sorry, SARS. SARS has no asymptomatic people. So it is twice arose, both times in China. And one time it actually got into Canada and the United States, a few people, but they were able to isolate them and quarantine them. And then SARS, which first became spreading in the world in 2003, they got it to go extinct, at least in people. And then it later popped up again in China, and the Chinese quarantined everybody who was sick, and it didn't spread beyond China that second time. So that was an easy way to control SARS, and that is everyone who was infected had symptoms. That's not the case of COVID-19, and how COVID-19 entered this country was an asymptomatic patient brought it into this country. If you remember, Trump actually put on a quarantine. Nobody from China could enter the country. And then later that was added, I think nobody from Italy could enter the country. And maybe he expanded it to Europe, but still COVID-19 entered the country. And the reason is because COVID-19 patients can be asymptomatic. When we're talking about young people like you guys and children, about 50% of patients who get COVID-19 can be asymptomatic, where they have no symptoms, no sign that they are ill. COVID-19 had a mortality rate something between 0.2% to 3%. That now has dropped to about half that numbers. And that's because COVID-19 has become less lethal. So it's about half as lethal as it used to be. But it does spread more easily than the flu. And one person is likely to infect 1 to 2.5 other people, which is why COVID-19 has continued to be a problem in the human population. COVID-19 does have a scientific name. It's called COVID-19 because it's a coronavirus. And it was ID'd in 2019. So COV for coronavirus, ID identified 19 in 2019. And 2019 was when COVID-19 first appeared in people in China. It does have a scientific name, Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-Virus 2. And that's so similar to SARS that even scientists tend to call it COVID-19 to distinguish it from SARS. COVID-19 is closely related to bat coronaviruses, and it likely originated from bat coronavirus. They've actually sequenced the COVID-19, the different strains of it, and they discovered the first strain hitting China was a combination of two bat coronaviruses that recombined with a third coronavirus that came from a pangolin. A pangolin, another name for that, is a spiny anteater. So this was a virus that recombined not once, but actually twice, because 3 viruses recombined to make the COVID-19. It is likely that a pangolin was an intermediate for this virus and then brought the virus to humans. And the virus was likely brought to humans in the Chinese food market because the Chinese had all sorts of exotic animals in there for consumption, including pangolins. By the way, the Chinese have shut down those markets and I think they still have the food market, but it's only normal food. They do not have all of the exotic animals for sale for people to eat, because 3 times in these Chinese food markets, we have gotten a serious disease coming out of them. COVID-19 was actually the last. The second was the second case of SARS came out of the Chinese food market. And the first time was also SARS in 2003, when SARS first appeared. And the Chinese came to the realization that having all these animals crowded together with a bunch of people tended to bring new viruses, which could be lethal to the human population. And so they've cut down at least the number of animals that they're selling in the Chinese food markets. I believe they still have, like, cattle and beef and pork and things like that. They still have the food market. They just don't have the exotic animals. Any question about any of that? All right. The transmission of COVID-19 is primarily via droplets from coughs and sneezes within a range of 6 feet. However, it may spread from other ways, like simply breathing, or singing is another way the COVID-19 likes to spread, and certainly talking. And it may spread longer than 6 feet, but generally within 6 feet is the greatest risk. It can also spread by contaminated surfaces, which is kind of odd that COVID-19 doesn't tend to spread more that way. It doesn't like to get on a surface and then infect our hands, and then we self-inoculate ourselves, because that is actually a common way that we get colds and flus. We get the virus on our hands, and then we inoculate a mucous membrane in our face with the virus. And that's a way we get a lot of colds and flus. With COVID-19, we can get infected that way, but the virus just doesn't like to travel that way. It likes to travel directly from person to person. So it can, like, for example, get on mail, and then if you touch the mail, you can pick up the COVID-19. That is possible, but it's usually breathed in and rarely picked up in other ways. It can survive for 3 days on plastic or certain metal surfaces, but it can only survive 1 day on cardboard or paper. So what I do is I always just hold my mail for a day before I open it. The other thing you could do is open your mail and alcohol your hands if you want to be COVID-19 safe. The symptoms, COVID-19 is mostly flu-like. The runny nose and sneezing are a little less common than the flu, and then the shortness of breath and the deaths are more common. Mortality increases with age of the patient, with the ill health of the patient. It's more common in men than in women, and is more common in blacks, and Hispanics than it is in whites and Asians. Why actually men and blacks and Hispanics has never been really explained. It's just the way the virus is. The incubation period of the virus is 2 to 14 days. So you have to be exposed to the virus for 2 to 14 days before you get a sign or symptom. And then a lot of people, especially young people, are asymptomatic. The estimated death rate, the last I checked in the United States, was 1.8% of the patients getting COVID-19 died. That's probably decreased now because the virus has changed and is less lethal, but it still is lethal. Prevention, how do you prevent getting COVID-19? Use correct hand washing. If you touch something that could be contaminated, you wash your hands. Keep your distance from people, especially if they're coughing or sneezing, and then don't touch your face. If you're sick, you should wear a mask to prevent the virus from spreading, and it's not a bad idea to wear a mask to also prevent the virus from getting to you. It's best to stay home if you're sick and cough into your tissue or your elbow. Any questions about COVID-19? All right. If not, I'm going to stop there and we will pick up here next time. Let me look quickly about the lab. I don't think we're going to have a lab today. Yes, no lab for today. So I will be there for 15 minutes to answer any questions. After 15 minutes, I'm going to log off. Any questions? All right, I'll see you on Thursday then.